Hello, everybody. We are going to get started here in just 1 second. We're letting everybody in. Thank you for joining us this evening. We're so happy that you're here. I am Allison Foss, and I am the Executive Director of the Myasthenia Gravis Association. Again, if you're new here, we're really happy that you're joining us this evening. After the presentation today, you'll get a survey. We would love your feedback. One other quick note, I wanted to let you know we are doing a webinar on Monday. Our virtual monthly meetup will be from 6:30 to 7:30 p.m. There will be a link in the survey going out where you can sign up. So we hope we'll see you on Monday as well. After today's presentation, our comments and questions will be answered at the end of the presentation today. So I wanted to introduce you to Dr. Jeffrey Guptill. He is the Neuromuscular Franchise Lead in Clinical Development at argenx. In his role, he oversees the late-stage clinical development for programs for tenured, sorry, I always butcher that, Vyvgart, and Ken, I'm going to butcher the second one, the C2 complement inhibitor. He is also the Adjunct Associate Professor of Neurology at Duke University School of Medicine. Dr. Guptill received his medical training at Virginia Commonwealth University and subsequently completed an adult neurology residency and neuromuscular medicine, EMG, and advanced neuromuscular disease fellowships at Duke. Prior to joining argenx, he was a tenured Associate Professor of Neurology at Duke University. Dr. Guptill, I'm going to turn it over to you.
Great. Thank you, Allison. And thanks for everyone who's attending this evening and for spending some of your evening with me. So today I'm going to be sharing some information about a clinical trial that is actively enrolling currently. And Allison, you can see the slides still, right? Just want to double check.
Yes, we can.
Okay, great. All right, so we will talk about this study called ADAPT SERON, and this is a study that's being conducted by the company called argenx that I work for. And this study is for patients with acetylcholine receptor antibody negative generalized myasthenia gravis. So it's patients that don't have the most common form of generalized myasthenia with AChr antibodies. And right off the bat, I just want to emphasize, because this is one of the most common questions that we get about this study, is does it include patients that have MuSK or LRP4 antibodies? And the answer is yes. So this study is designed for patients that either have MuSK antibodies, LRP4 antibodies, or those that have no identifiable autoantibodies. So really the group that is not part of the study are the patients that have AChr positive generalized myasthenia. And we'll talk about this in more detail as we move along. So first I want to start by talking about what is a clinical study before we get into the details of this particular study, the ADAPT SERON study. So a clinical study or a clinical trial is conducted to learn whether an investigational drug is safe and effective for patients. And these studies are done under strict standards to ensure that the best data is available at the completion to inform healthcare decision-making and the decision-making of regulatory agencies that decide whether or not to approve a drug. And so that would be like the Food and Drug Administration, the FDA. So as a result of this, these studies are done following a protocol, and the protocol describes the type of patients who can participate in the trial, the schedule that the participants and the investigators and the study teams must use to conduct the different tests and procedures that are part of the study. There's a lot of detailed information about the investigational drug that will be evaluated in the study, and the dosages, and as well as the length of time that the study will take. How long will someone need to be in the study to participate? And of course, like, we also have to define what are the objectives of the study? What are we trying to learn from it? And this is all described in the study protocol. And so oftentimes this leads to the question why this is important. And so these types of studies are really critical to aid in the understanding of how a new therapy impacts patients. Is it safe, and is it effective? And this is really important information to know before it's, put out there more broadly for patients to have access to it. And as many of you know from your experience, is that a lot of the treatments that are used to treat myasthenia gravis traditionally have not been approved by a regulatory agency, and they've all been used off-label, so to speak. So in addition to having a regulatory approval, hopefully for this product in the seronegative group of patients, we are also doing the study in a way that will contribute to additional medical knowledge, and that will hopefully lead to improved patient care through generating appropriate evidence that healthcare providers and their patients can make the best decisions about their healthcare in the future. So for this study, we will be using a drug called Efgartigimod alpha. Efgartigimod alpha is not approved by the FDA not approved by the FDA for the treatment of AChr antibody seronegative generalized myasthenia. And of course, the purpose of the study is to evaluate whether it's safe and effective in this population. It's important to note that, unlike some other investigational drugs that haven't been studied in patients with myasthenia before, efgartigimod is approved by the FDA for the patients of AChr antibody positive patients, and many of you may be aware of that. But of course, this is a different subgroup of patients than those that have seronegative GMG, and that's why it's important to do the study. Efgartigimod, the way it works is that it lowers IgG. So IgG is our antibodies, essentially, and it lowers all classes of IgG molecules. And the concept is that it also reduces the pathogenic or the harmful antibodies that attack the muscle in myasthenia. And so in the case of AChr positive patients, it lowers the AChr antibodies, and this is associated with improvement in the patient's condition. And the hypothesis is that doing so in a MuSK antibody positive patient or LRP4 positive patient, lowering their antibody levels translates to hopefully improvements in their clinical condition. For patients that don't have antibodies, the assumption is that the patients have antibodies that just aren't detectable with the current assays that we have available to us. And so it's still the same concept that it's an autoimmune disease, and that by lowering these pathogenic but undetectable antibodies in the blood, that we can improve the patient's condition. So next, I'll talk a little bit about AChr antibody seronegative GMG before we get into more details on the study. So some of this is probably familiar to many of you, but just to make sure we're all on the same page. So generalized myasthenia gravis results when the body's immune system starts attacking its own muscle membrane. And this disrupts the nerve to muscle connection, resulting in the clinical symptoms that patients experience of muscle weakness, fatigue, and difficulty with muscle control. Many of you are familiar with the fact that there's a wide spectrum of severity of myasthenia gravis, which can range from very mild symptoms like drooping of the eyelids to very severe, debilitating, and often can be life-threatening symptoms. So most of the patients have acetylcholine receptor antibodies detectable in the blood. But there are a subgroup of patients that don't have these antibodies, about 20% of them. And within that group, it's heterogeneous. There are several different subgroups within the AChr negative population. There are some patients that have MuSK autoantibodies or LRP4 antibodies, and then some patients that have no identifiable antibodies. And as I mentioned earlier, all of these patients are the population that we're hoping to study in this new trial. Currently, the treatment for seronegative MG is very similar to what is done for AChr positive patients, and that is to help improve the communication between the nerve and muscle. And this is typically done with medications that either are symptomatic treatments like cholinesterase inhibitors, mestinon or pyridostigmine is one that you're probably familiar with that almost all patients will receive at some point. And then other immune suppressing or immune modulating medications like steroids or mycophenolate, for instance. For AChr positive patients, there can sometimes be a surgical intervention or thymectomy that can sometimes be done. The issue that we have with seronegative MG is that with a lot of these medications, the patients haven't been as well studied as the AChr positive group of patients. And so we don't have as much information on what works for this particular group of patients. And so we need more, in general, we need more studies out there that are evaluating the benefits and risks of the different treatments that can be given for patients with myasthenia gravis. And so argenx is interested in conducting this study with efgartigimod to learn more about its effect in seronegative MG. So let's talk about the study a little bit. So the study is called ADAPT SERON, and it's a global clinical trial. And again, to emphasize, it's for a broad spectrum of patients with seronegative gMG, so patients that have MuSK, LRP4, or no identifiable antibodies. There are 22 countries that will be participating, and 18 sites will be in the United States. And this study will be done to compare the efficacy or whether a patient can improve from their background therapy using either efgartigimod or placebo, which is basically a water infusion. But in a situation where neither the patient nor the study team knows which product the patient is receiving. So no one really knows who's getting efgartigimod or who's getting placebo. And this is called a placebo-controlled clinical trial. And this is considered the most rigorous way to study a new treatment. On the right-hand side of the slide is a QR code that you can use your camera on your phone for if you want to learn more information about the study. You don't necessarily need to click on it now. At the end of the presentation, we'll also have that QR code and a web link as well, and some other resources if after this presentation you're interested in learning more. So now I'll give you a little bit more detail about the study itself. So an important question for many people who may be considering a clinical trial is, well, how long is it? And so this study is 2 years and approximately 2 years and 2 months. And during the study, a participant will need to make regular visits to a trial site for evaluations and administration of the investigational drug or placebo. And for those of you who aren't familiar with placebo, like I mentioned it as a water infusion, but it's basically another investigational product that looks exactly the same as the test agent, Efgartigimod, in this particular study, but doesn't contain the active ingredient. So it's as close to the real thing as you can get, so that no one knows which it is. So within the overall 2 years and 2 months of the study, it's broken up into 3 different parts. So there's a screening period, which is to verify eligibility, and this lasts up to 5 weeks. Then there's a part A, and this is the part where it's placebo controlled, where the participant either gets Efgartigimod or placebo, and this lasts for 8 weeks. And then after that, there's a part B, which lasts up to 2 years. And in part B, it's called open label. And in that part of the study, everyone gets Efgartigimod. So I think one of the potential nice things about this study is that the placebo-controlled period of the study is relatively short. And this is, so prior to me joining argenx, when I was in clinical practice and doing clinical trials, this was a common concern of patients was, well, how long am I potentially going to be on placebo and not getting the real drug? That people were really interested, of course, in trying the new drug, and wanted to make sure they also had the opportunity to try the new drug. And so for this study, it's relatively short compared to a lot of other trials that are out there, where it can be 6 months or so, or 3 to 6 months, where you're on potentially on placebo. In this study, it's 8 weeks, and then after that, you would a participant transitions to treatment with Efgartigimod, where everyone gets Efgartigimod. And I say that the study is up to 2 years and 2 months, but of course, like every participant has the right to withdraw from the study at any time and for any reason. So it's not like you're stuck in the study if you agree to participate. Of course, we want people to participate for the full duration so we can learn the most from them, but we understand that people's feelings can change, or their situation can change. Like, for instance, I've had times where a patient moved to another country or another state and they were unable to participate any longer, and that's just an example of why someone might need to stop participating in a study. So let's go over the study design a little bit. So during each of the visits, patients will have an examination and assessments to monitor the effect of the drug once they're in part A and part B, and to see how they're experiencing the investigational medication. At the end of the part A, which is a placebo-controlled part, as I mentioned, patients can then move on to part B of the study, where they receive open label treatment. So I think this slide is helpful because it I think at least for me, I'm a visual person, so it helps me to understand the setup of the study. And so I'll just walk through this for you. So I mentioned the 3 different parts of the study. So the screening period, part A and part B. So the screening part is to assess whether a participant who's interested in the study is actually eligible based on the requirements for the protocol. So, for instance, people will be checked to see if their disease is severe enough to be part of the study. The study is looking, for patients who have evidence of active disease and that have generalized symptoms, meaning that involves their arms or legs or their swallowing or breathing. So patients that just have ocular symptoms would not be eligible for this particular study. We'll also look at the other medications that a patient may be receiving, and look at laboratory work to ensure that it's safe for them to also participate, that there are no, expected interactions or problems with other medications or other underlying conditions that would make it unsafe for them to participate. So after the screening period, if a patient is eligible, they would then move to Part A. And we're expecting about 110 participants in the study will be randomized. And so at this point, at the beginning of Part A, patients will be randomized to either receive efgartigimod or to placebo. And what randomization is, it's basically there's a computer system that assigns whether a patient gets placebo or Efgard. It's kind of like flipping a coin. And so a participant will end up either with efgartigimod or placebo. They'll get 4 doses of either the placebo treatment or efgartigimod, and then they will be followed for 5 weeks after that. And then at the end of that period, then the participant will transition to open label treatment. So if they were getting placebo, they would transition onto efgartigimod. If they were getting efgartigimod, they would just continue getting they would continue getting efgartigimod. And at first, in the Part B, the dosing will be given on kind of a fixed schedule to get everyone off on the same cadence. And then after that, after the first couple treatments, then the investigators will have the opportunity to potentially space or adjust the timing of each cycle of treatment based on the patient's needs. So what we found in the AChr positive group of patients is that some patients need treatment like every 4 weeks. Others can go 8 or 10 or more weeks in between treatments. And so we have this adjustability to provide treatment based on the need of the individual patient. So during Part B, the participants will still need to come back to the clinical trial site to have regular follow-ups to make sure that they're not having any problems in the study, and to see how the treatment is impacting their myasthenia gravis. In Part A of the study, the assessments are quite frequent. They're basically weekly for those first 8 weeks. And that's because that placebo control part is really, really important for understanding the relative benefit of efgartigimod versus the placebo treatment. And then after that, in the open label period, the visits tend to space out more over time, and it becomes a little more convenient for patients. There still is a commitment, though, that participants will need to come back, more so than they would for a regular, clinic. If they were just being followed by their clinician, it's typically more than would be expected for that. But it's something that you can talk about with your treating physician. Okay, so we will move on to the next part of this. Okay, so and then within each type of visit, there are basically 2 different types. There are treatment visits where the participants will receive the study treatment, either active or placebo. And then there will be follow-up visits where they're just being assessed to monitor their symptoms and signs of myasthenia, as well as monitoring for any side effects. During the treatment visits, you'll get so during Part A, placebo or efgartigimod over the course of an hour, and then it's followed by a 30-minute observation period. And then in Part B, everyone will get efgartigimod again over an hour with a 30-minute observation period. Oh, I knew there was one thing I knew I was forgetting. So another thing I wanted to mention here is that During this study, participants will also be able to stay on their current background treatment. So it's not like to participate in the study, you have to come off of your treatments like pyridostigmine, or if you're on prednisone, that you don't have to come off. This is added onto the background of your treatment. But the main requirement is that just your treatment needs to be stable before you can enter the study. And so, there is specific language in the protocol that addresses how long different medications need to be stable before participating in the study. Okay, we covered that. So that's the highlights of the study itself. Now, what follows are some kind of frequently asked questions, I guess you could say, that we'll go through here, and then of course at the end we can also take any other questions that you have. And some of these we've probably already addressed already. So first of all, like, what is the purpose of the trial? So the purpose is to look at the efficacy and safety of Efgartigimod IV compared to placebo in patients with AChr seronegative GMG. And we also want to look at the long-term efficacy, safety, and tolerability in the open label period during Part B. So the open label period gives us more information about the long-term effect on how patients do over up to 2 years. So why do we need this trial? So hopefully, as I mentioned earlier, convinced you that these types of studies are really important from getting approval from the FDA, and also to increase our understanding and knowledge of seronegative MG, as this population has been excluded up until very recently from clinical trials. And currently, in the US, there are very limited evidence-based options for treating seronegative patients. So hopefully we can provide some new information on this. So who qualifies? So as I mentioned, we're trying to enroll about 110 patients, and it is a broad population of seronegative MG. So generalized MG patients that have either MuSK, LRP4, or no identifiable autoantibodies. I mentioned earlier that the study is global, and so we want to have broad representation across globally of patients with MG. This helps us to understand whether there are different effects in patients or participants with different characteristics, men versus women, and different autoantibody status, etc. So how will efficacy be evaluated, or put another way, how will we know if it works? So I mentioned earlier that the real critical part of this study for determining whether it works is the Part A, the placebo-controlled part of it, where the patients don't know and the study team doesn't know whether a patient's getting Efgartigimod or placebo. And during Part B, everyone will gets Efgartigimod. So the efficacy of Efgartigimod versus placebo, the two primary measures that will be used are the MG-ADL and the QMG. The MG-ADL, some people may be familiar with already. So it's a questionnaire that assesses the impact of GMG on daily functions, and this score will be monitored over time from the patient's baseline at the time that they start the study to the end of the Part A. The second scale is called the QMG, which is a standard scale that's done by an evaluator, and it measures like strength and fatigability of various muscle groups, and this outcome measure has been used in many MG clinical trials. It's not something that's commonly done in the clinic, though, because it's a little bit more involved. So another common question is, will I receive study medication? And so to emphasize this again, in part A, half of the participants will get Efgartigimod and the other half will get placebo. But in part B, everyone will have the opportunity to receive Efgartigimod. And both of the medications are administered in an IV. Okay, this is an important one. What about my privacy? So, you know, for these clinical trials, there are sets of regulations that exist to do the best we can to maintain patients' privacy within a study. And so it's expected that the study doctors and staff at the site, as well as the company that's running the study and all of the other people that may be involved, are treating each participant's health information in a confidential manner. One of the main ways to do this is to give each participant a study number. So your name is not used, for instance, in terms of in the study records. So each participant will have their own unique study number, and that will be used to collect information to maintain patients' privacy. And really only the site where at the clinic will have the link between the participant and their study number. So what else should I know? So of course, this is a clinical study. We don't know for sure whether Efgartigimod will help patients with seronegative MG. That's why we're doing it. So if someone participates in the study, it's possible they could stay the same, they could improve, or they could get worse. But we wouldn't be doing the study if we didn't think that there's a potential that we can help patients with this. And the fact that AChr positive patients have already benefited from Efgartigimod also gives us some confidence to move forward with a study like this. And of course, the unmet need that patients have. So in addition to evaluating this study to gain an approval for the drug, we will also participation in this study also helps to improve our knowledge within the overall field of GMG, and specifically the seronegative population, so contributing to the medical knowledge. So participants also like to know if it's going to cost them anything to participate in the study, and the answer to that is no. And all of the study procedures and assessments are the participant does not have to pay for. In addition, we know that because MG is a rare disease and there aren't a lot of centers that specialize in treating MG, that a lot of patients travel long distances to be able to see specialists who focus on myasthenia gravis. So as a result of that, we want to reduce the barriers to participation in the study, and so we are also will pay for the travel expenses for a participant. We recognize that we're asking a lot for participants to make multiple visits over a short period of time to the trial site to participate in a study. And we want to try to alleviate some of that burden. And one way that we can help with that is with providing travel expense reimbursement. And some study visits we may also be able to take place at home with a qualified study nurse, which is also a measure to try to reduce the burden for participants. So that's about the end of the presentation. So if you have interest in learning more about the study, this slide can give you some more ways to learn more. You can use the QR code on the right. There's also the website called adaptseron.com. And there's also a phone number that you can call to learn more information. So with that, I will Oops, sorry, I went the wrong way. I will end the presentation and I will open it up for any questions that any of you may have.
Hey, thanks, Dr. Guptill, for that presentation, and thanks to Allison and the MGA for letting us join this evening. We do have a couple of questions in the chat and also in the Q&A. So the first one is, why was the study designed to give cycles of 4 weekly treatments and not weekly continuous treatments?
This is a great question. Yeah, so the original studies, the pivotal studies that led to the approval of Efgartigimod for generalized myasthenia for patients that had AChr positive MG were done with this cycle-based approach. And the concept here is that we recognize that every MG patient is different, and the treatment requirements for every patient is going to be different. How much, for those of you who are on existing standard of care treatments, if you're taking prednisone, some patients require high doses of prednisone. Other patients require low doses of prednisone. Some require nonsteroidal immunosuppressant drugs like mycophenolate or azathioprine. Others don't. So this study design is done in a way to provide the maximal flexibility to patients and their neurologists in terms of how to treat their myasthenia. So although we have the initial period stage A that is there, everyone gets kind of the same treatment up front. In stage B, the treatment can be adjusted based on the individual patient's needs. And so we think that this is advantageous to patients rather than just putting everyone on the same amount and interval of treatment without having to think about it or to adjust based on patients' needs. Great question.
Thank you. We have another one, a couple of more coming in the Q&A. So this one is, my flare-ups occur in cycles 2 to 3 times a year. Each symptom remission cycle includes increasing and decreasing prednisone in conjunction with the symptom severity. How do you differentiate the effect of prednisone to reduce symptoms with the efficacy of the drug?
Okay, this is another great question. So yes, so I mentioned at one point in the presentation that patients can come in on their existing treatments, and that the key thing is that their treatments need to remain stable during the course of the study, and particularly during stage A. So during stage A, we don't allow any changes of their medications for the exact reason that was mentioned, or for the exact problem that the person mentioned in the Q&A, is that if we allow a change in the dose of their prednisone, then we won't know whether if a patient got better or worse. We don't know if it was related to their Efgartigimod treatment or placebo, or was it related to their prednisone. So for that reason, during that period of time in stage A, we really don't want any changes to their treatment, so that we can distinguish if a patient gets worse, that we can attribute it accurately to, is it placebo or Efgartigimod or something else. Yeah, so I guess the other important point on this is that this doesn't mean that if a patient isn't doing well, if there's a problem in the study, that they're out of luck and that they're in a bad position. There are also considerations in the protocol for what to do if a participant gets worse. And so there is a provision for providing a rescue treatment if a participant needs it, if they're experiencing any problems with their MG.
It's on the same thread of treatments. Would IVIG, is that prohibit a patient from being in part 1 or being in the study?
Yeah, so there's a washout period for IVIG before they can participate. I believe it's 4 weeks. So you have to be like 4 weeks from your last treatment with IVIG or plasma exchange, and then you can enter. So, and again, this is for the exact same reason, is that if a participant comes into the study, they got IVIG, and then the next week they start Efgartigimod, we won't know if they got better. Was it because of their IVIG or was it because of Efgartigimod? So we need to leave some time there for the IVIG effects to kind of wear off.
So looking at the treatment of Efgartigimod, why did we go with the IV instead of sub-Q for this study?
Yeah, you guys are great. These are awesome questions. So, yeah, so this was we had a lot of conversation about this in the team, as to whether to do this study with the IV or the subcutaneous formulation. There are some specific reasons related to regulatory requirements that really pushed us in the direction of using the IV formulation. So number one is that the original study in generalized MG patients with AChr positive disease was done with the IV formulation. And so by doing this study also with the IV formulation, we can we have a better opportunity to compare the effect of the clinical effect in the seronegative and the AChr positive patients. Because there was a placebo group in both of those different studies. We wouldn't have been able to do that with the SC formulation.
Thank you. We've got another question about immune deficiency, such as common variable immune deficiency. Would that disqualify someone from the study?
So yeah, so there isn't a specific exclusion criteria for this. There is a general eligibility criteria related to basically anything that puts the patient at an increased risk to have a problem. Basically, that a medical condition that would make it relatively unsafe for a participant to be in the study. And a lot of this is at the discretion of the treating physician. So the physician at your site that's treating your MG would need to make that decision. So if this study was something that was of interest to you, I guess I'd recommend going back to talk to your treating physician to talk about it more and to see whether or not it could make sense for you to participate or not. Yeah.
Yeah, another question, I think you've pretty answered it very, very similar to what you just said, is if I had a critical reaction to IVIG in the past and it had to be discontinued, would I still be able to participate in this trial?
Yeah, so I think that an alternative to that could be plasma exchange. I think the one thing we would need to need to be sure that was available to you or accessible where you are, because we know that not everyone has access to plasma exchange.
All right, that's all the questions in the chat and the Q&A. I don't know if there's any others. If not, Allison, we'll turn it back over to you.
I don't see any other questions either. So this was fantastic. Lots of good information. If nobody else has questions, we can go ahead and wrap it up for this evening. Anybody else? I think that's it, guys. Well,
Okay.
thank you so much for joining us this evening, and we hope to see you soon. Oh, hold on. Looks like there's one more question.
Yeah, so let's see it here. So if I'm between flare-ups on a relatively low dose of prednisone, then I must stay on that low level throughout the study. If so, then if I'm in the placebo group, then I must stop the study.
So if you're on placebo, on prednisone, and you enter the study, the dose of the prednisone would need to remain stable for as long as possible, for sure, right? But particularly through the part A, so that 8 weeks. Then after that, it's a little bit more flexible in terms of the medication treatment. Now, of course, like in part B, everyone will be getting Efgartigimod, so you'd be getting an additional treatment in addition to the prednisone. So, and I think that, you know, hopefully it would, you know, providing the Efgartigimod would make it, you know, so that you didn't have any more flare-ups. I mean, I think that's the ultimate goal, but obviously we can't, you know, predict that or say anything confidently whether that'll be the case, but that's the idea. Yeah.
All right. I think that's it. Thank you so much, everybody, and have a great evening.
Great. Thank you so much for your time.
Everyone.
Thank
you.
you.